Hello everyone, welcome to this presentation on enzymes, the biocatalysts of life. In this session, we are going to explore what enzymes are, how they work, and why they are absolutely essential for every living organism. So, get ready to dive into the microscopic world where chemistry meets life. Let's start simple. What exactly are enzymes? Enzymes are biological catalysts. That means they speed up the chemical reactions that happen inside our cells without being used up themselves. For example, think of digestion. When you eat rice or bread, enzymes like amylase in your saliva start breaking down starch into sugar right inside your mouth. Without enzymes, that same reaction would take hours, but thanks to enzymes, it happens in just seconds. Now, let's understand what enzymes are made up of. Most enzymes are proteins, big folded molecules made up of amino acids. However, some enzymes are made up of RNA. These are called ribozymes. Here is something cool. Every enzyme is specific. It acts only on one type of substance called its substrate. Just like a key fits only one lock, an enzyme acts only on one particular molecule. For example, lipase acts only on the fats and pepsin acts only on the proteins. And maltase breaks only maltose into glucose. So, enzymes are not just powerful, they are also incredibly precise. Now, not all enzymes are made up of protein alone. Some need a little help, and that's where the cofactors come in. An enzyme without its helper is called as an apoenzymes, and these are inactive form. When you add the helper, which can be a metal ion or an organic molecule, the complete active enzyme is formed. That is called a holoenzyme. Depending on how the helper is attached, we give it a different names. If it's tightly bound, it's a prosthetic group like FAD. If it's loosely attached, it is a coenzyme like NAD+, which comes from vitamin B3. And if it's a simple metal ion, it might be zinc, magnesium or iron. For example, carbonic anhydrase, an enzyme that helps you breathe efficiently, needs zinc to work properly. So remember, apoenzyme plus cofactor gives you holoenzyme, the complete active form. Now let's look closely at how enzymes actually work, the mechanism behind their catalytic power. Every enzyme molecule has a special region called the active site, a small pocket or groove on its surface. This is where the reacting molecule called substrate fits in. The active site is shaped and charged in such a way that only its specific substrate can bind. This is what we call enzyme specificity. Once the substrate binds, an enzyme substrate complex is formed, which will give enzyme plus product, where enzyme is unchanged, forming new product from substrate. Now, how does the substrate actually fits in? There are two classical models that explains this. First is lock and key model, which is proposed by Emil Fischer in 1894. In this model, the active site of enzyme is considered a rigid structure like a lock, and only the correctly shaped substrate, that is the key, can fit perfectly. For example, the enzyme urease fits only urea, not any similar compound. This model explains enzyme specificity very well, but not the flexibility. Second model is the Induced Fit Model, which is proposed by Daniel Koshland in 1958. Here, the enzyme is not rigid. When the substrate approaches, the enzyme's active site slightly changes its shape to fit the substrate more snugly, like a glove adjustment to your hands. This change brings reactive groups of the enzyme into the right position to stress or weaken the bond in the substrate, making the chemical reaction occur much faster. After the reaction, the product is released and the enzyme returns to its original shape, ready to catalyze again. Let's take a real life example to visualize this. When you eat something sweet, the enzyme sucrase acts on the sugar, that is sucrose. Sucrase binds sucrose at its active site, breaks the bond between glucose and fructose, and releases them as a simple sugars that your body can absorb for instant energy. So, in short, enzymes work by forming temporary enzyme substrate complexes. lowering the activation energy and converting the substrate into the product, freeing themselves unchanged for the next round of reaction. That's why we call enzymes the true catalysts of life. Now, let's see the characteristics of enzymes. First, they work under mild conditions, normal body temperature and neutral pH. For humans, most enzymes work best around 37 degrees Celsius. That's our body temperature. Second, they are highly specific. One enzyme works only on one substrate. Third, they are extremely efficient. For example, the enzyme catalase can break down about 1,000,000 molecules of hydrogen peroxide every second. Talk about the speed. And finally, enzymes are reusable. They don't get consumed in the reaction. So, the same enzyme molecule can work again and again. So, enzymes are like nature's ultimate multitaskers, fast, precise and tireless. Now, just like any machine, enzymes have ideal working conditions. Change those and they slow down or even stop working. There are 4 major factors that affect the enzyme's activity. First is temperature. Enzymes work best at optimum temperature. For us, that's around 37 degrees centigrade. Too low and reactions slow down. Too high and enzyme get denatured. They lose their shape and stop working. So, optimum temperature is very important for enzymes reaction. Next is pH. Every enzyme has its preferred pH. For example, pepsin in your stomach works best in acidic conditions because there is acid in your stomach. So, optimum pH for pepsin is pH 2, while trypsin in your intestine prefers an alkaline pH that is around 8. Next is substrate concentration. Increasing the amount of substrate increases enzyme activity up to a point called Vmax when all enzyme active sites are busy. Next is enzyme concentration. More enzyme molecules means faster reactions as long as there is enough substrate available. So in short, enzymes work best when the environment is just right. Not too hot, not too cold, not too acidic and not too basic. Alright then, that's all for today's session. We have explored the basics of enzymes, what they are, how they work and key factors that affect their activity. In our next video, we will continue with the remaining exciting topics, that is enzyme kinetics, enzyme inhibition, classification of enzymes and their biological and industrial applications. Those concepts will help you connect everything we learned today to real life systems, from your own digestion to biotechnology labs. So make sure to join me in the next part. Till then, keep revising, stay curious and remember, enzymes make life possible one reaction at a time. See you in the next class.